Hello everyone, today I'm going to present TAIL, transformer-based protein function annotation with joint sequence label embedding. So overall, my presentation includes 4 parts. So first, I will go through the introduction, and then I will describe our method. Thirdly, I will talk about the results. And finally, I will end my presentation with conclusions. So first, for the introduction part. So why we need protein function annotations? So basically, understanding protein sequence function relationship can provide enormous benefits for biological and pharmaceutical applications. However, there is a significant gap existing between the availability of sequences against the availability of their functions. For instance, there are a large number of Trumbull sequences that are waiting for annotation. However, only a tiny fraction of them have manually annotated sequences in Swiss plot. In order to close such gap, there are a lot of previous work. Basically, they can be classified as homology-based methods such as BLAST, SciBLAST, and Diamond, or machine learning-based methods. So in machine learning-based method, protein functions can be predicted either from sequences, structure, PPR network, or literature text. Also, the protein function can be predicted from the multiple sources combined together. However, as we know, only around 0.01% of sequences in Trumbull have solved those structures in protein data bank. Of course, if one considers homology models or even structure information such as domain, family, or motif, such number can be much higher, but yet still a very low value. Moreover, 70% of sequences in Trumbull have available network information in StringDB. That means there are still a large portion of sequences in Trumbull have only sequence level information. So this is why we need a sequence-based protein function annotation, which is also the focus of this study. So what we are focusing on is even tougher cases. Specifically, we are focusing on the sequences in the twilight or even midnight zone, where homology-based methods usually fail, because in Kappa, performances on many methods deteriorates fast when sequence similarity decreases below homology levels. Moreover, we are focusing on sequences with some specific function labels that are never or rarely seen in non-annotated sequences. Because deep learning is actually a data-hungry technique, and many methods often have to get rid of a large number of geoterms with few annotations, leading to narrow applicability. For instance, some methods have only considered 5,000 geoterms with at least 50 annotated sequences each, which only accounts for less than 12% of all geoterms. While in our case, we consider all geoterms with at least 1 annotated sequences, and that actually accounted for 64% of all geoterms. Therefore, to summarize our contributions, first, we proposed a transformer-based architectures which can deal with the long-term dependencies among protein sequences compared to CNN, so that we can have better generalizability. Secondly, we propose a joint sequence label embedding framework that can deal with the tail label situations. And thirdly, we propose a hierarchical regularization term that can better reduce the hierarchical violations. Okay, then I will talk about our method. So overall, this is the architecture of our method. So actually it includes 4 components: a sequence encoder, a label embedding, and a joint sequence label embedding, and fully connected and output layers. So I will go through each component one by one. So first, for sequence encoder, it actually consists of 2 parts, a sequence embedding layer and a transformer encoder. So for every input protein sequence, it will convert it into a latent state with dimension n plus h, where n is the length of the sequence and h is a hidden dimension. So besides the sequence encoder, we also have a label embedding to embed every label in the gene ontology graph. So it will embed a gene labeler matrix into a latent space, into the latent state with dimension c times h, where c is the number of labels and h is a hidden state. After that, both the sequence hidden state and the label hidden state will go into a joint sequence label embedding framework. And this framework consists of a matrix multiplication layer, a 1D convolution layer, and a max pooling layer. The output of this joint sequence label embedding framework will going back to reweight the sequence hidden state. And such reweighted hidden state will go to a fully connected layer and eventually go into an output layer. And actually in the output layer, we use a sigmoid function to convert to the predicted score into the predicted probability so that it can be between 0 and 1. So besides architectures, in order to mitigate the hierarchical violation, where the predicted score of a child geoterm may be larger than those of its ancestors, we propose the following cross-entropy loss plus a hierarchical regularization term. So here L prime is actually the standard cross-entropy loss over all the training examples. And besides, we have a hierarchical regularization term R here. So this R is actually the average over a ReLU function over all the ages in the gene ontology. So this ReLU function actually describes that if the predicted score of a child term is bigger than its parent term, then we're going to penalize it. So until here, we describe all the components of tail. However, similar to DeepGo Plus, we actually use a convex combination of multiple tail models and diamond scores. So you can see here, the first term here is a simple average of multiple tail models, and the second term is a diamond score. So we use a convex combination to get our final model, tail plus. So K here is actually the number of tail models with different hyperparameter settings. So in most of cases, K here is tuned to be 3 based on the validation set. Then I will talk about the results. So for the data set, in this study, we're considering 2 data sets. The first is a kappa-3 data set, which include a training and validation set of the experimental annotations between September 2016, and a test set with experimental annotations between September 2016 and November 2017. Then, secondly, we have created our own data set, which has more sequences compared to kappa-3, and also we have more latest annotations. So first, we will show the results on kappa-3 test set. So here, we compare to several methods, including naive, which is the simple frequency-based method, diamond score, alignment-based method. DeepGo is actually a deep learning-based method. DeepGo CNN is an upgrade version of DeepGo, and DeepGo Plus, which is the convex combination of diamond score and DeepGo CNN. And eventually, GeoLabeler, which is the state-of-the-art of kappa-3, they're using learning to rank. So as you can see here, tail plus actually shows the superior performance on BPO, CCO over Fmax, and on all the 3 ontologies over AUPRC. So it actually reached the second best next to GeoLabeler on MFO over Fmax. So notice that GeoLabeler use curated features from sequence as input, while in tail plus, we just use a simple sequence alone. Then we see we compare different methods on our data set. So notice here, we replace GeoLabeler with NetGo, which is an upgraded version of GeoLabeler using network information. So from number here, we can see we can have the similar conclusions as in the kappa-3 data set. So thirdly, we actually compare different methods on the test set, on the portion of our test set without network information. So you can see here, tail plus actually reached the state of the art on all the 3 ontologies across 2 metrics. So that means when network information is not available, TEL plus is a state of art. So for the second part of our results, we actually focusing on the generalizability analysis, because practically function annotation is needed the most in the twilight or even midnight zone. Therefore, we examine our models and the competing ones in their generalizability from the training set to the test set from 3 perspectives: sequence, species, and labels. First, we compare different methods in sequence. So in order to do that, we split our test set into 4 bins based on the maximum sequence identity against the training set. So here we compare TEL and TEL plus against the naive, diamond, typical CNN, typical plus. So the bar here is actually the value of Fmax of different methods on this bin, on each bin. And also we give the count on each bin in this gray dot. So the figure from left to right is actually on molecular function, biological process, and a cellular component. So as we can see here, in the low similarity region, the alignment-based method diamond score perform poorly, which is understandable. And also TEL and TEL plus perform, while TEL and TEL plus actually perform best. So that means actually our TEL and TEL plus shows very superior generalizability to proteins of low similarity. Secondly, we compare different methods on the new species and also low similarity, where the MSI is below or equal to 40%. So moreover, we split those subsets over eukaryotes and prokaryotes. So as a result, over these 6 bins, we can see TEL and TEL plus actually shows the superior performance here. So that means TEL plus shows superior generalizability to proteins of new species compared to the training data. And finally, we show the different models' performance based on the labels. So here, similar to the sequences, we split the test set into 4 bins based on the average label frequency, which actually define the label similarity of each test sample against the training set. So as we can see here, for this left bin, which means the low label similarity here, TEL and TEL plus actually shows the superior performance against all other methods. Then that means our methods has shown the superior generalizability to proteins of really annotated functions compared to the training data. So additionally, we actually perform an ablation study. So we start from the typical CNN and incrementally add our algorithm component. So for B1, we just replace the convolutional layer in typical CNN with a transformer encoder plus input embeddings. Then from B1 to B2, we replace the post-training corrections of hierarchical violations in B1 with the additional loss terms of hierarchical regularization. Then for B3, we just add the label embedding and joining similarity modules to B2 for joining sequence label embedding. So as you can see here, not only on the overall performance, but on the 3 subsets with different generalizability perspectives, every algorithm component contribute non-trivially to the performance improvement. Okay, finally, I will go into the conclusions. So in this study, we introduced a novel transformer-based deep learning model named TEL with joint embeddings of sequence inputs and hierarchical function labels. In addition, we use TEL plus, a convex combination of TEL and a similarity-based method diamond to further improve model performances and generalizability. Our results actually demonstrate that TEL plus outperforms all sequence-based methods in all 3 ontologies and outperforms the state-of-the-art sequence plus network method in BPO and CCO. When network information is not available, TEL plus outperforms all compared methods in all ontologies. Importantly, both TEL and TEL plus showed superior generalizability to sequences of low similarity and in never annotated new species, as well as rarely annotated functions. echoing the rationales of our algorithm development. Finally, in ablation study, we indicated that the improved performance is actually attributed to our newly introduced algorithmic components. So for the availability, our paper link is here. And for the data, trained models, and the codes, the link is here. Okay, thank you all for listening.